Hey, yo, what up? Mr. Rutherford here. I am going to share my screen here, and we're going to go over some AP Bio Unit 6 business. Now, I call Unit 6 evolution. They call Unit 6 gene expression regulation. Great review. Obviously, genetics, the gene is the connection between first semester and second semester. So we have really the DNA connection to evolution there. So, evolution is for them, they call Unit 7 natural selection. So whatever, it's great freaking review. So yeah, let's go over the evens here. All right. We have arsenic is toxic element. Yeah, blah, blah, blah. I'm going straight down to the question. Okay. Researchers claim the bacteria live in environments heavily contaminated with arsenic. So let's pick out the words we should focus on. When you read through this, okay. Okay. A conversion. This is a classic thing that occurs in biology. We have genes that encode for enzymes, which do a reaction, which means that there's going to be some sort of like chemical process. And this chemical process here is converting arsenic into arsenite and removing this toxin from their cell. Okay, great. So I'm going to like write that on my scrap sheet of paper that I always need to have out, this conversion thing that is going to occur. Okay, it's so easy just to write arsenic. I'm going to write non-toxic, and right next to non-toxic, I'm writing arsenite. Okay, and that is done by bacteria, right? It's not telling us. Okay, now let's go to the answers. There are multiple operons controlling the production of proteins that process and remove arsenite from cells in both these, okay? However, E. coli only has one operon, okay? So I'm looking up at this going like, okay, now we got to talk about the key thing that I saw was researchers claim bacteria can do this stuff, okay? So up here, what I'm looking at is E. coli is a bacteria. This other Hermanonanus, arsenate, but whatever, that's a bacteria. It says it right there, bacteria found. So you just need to understand we're talking about 3 different bacteria. Now, up in the question here, it says 3 different bacteria. So I'm just scanning this just to kind of check, like, okay, am I looking at this the right way? We have different bacteria, right? And then this word operons, that means we're talking about genes, we're talking about gene expression. And so that's just kind of like what the words I'm zooming in on to kind of get oriented on this, okay? So it says that the E. coli only has one operon. So up here, I'm looking like, okay, yeah, that's one operon. They're testing your knowledge that like that's one operon. That looks like a lac operon, looks like a trip operon. There's going to be 1, 2, 3, 4, 5 different genes, and those would be protein subunits that after they're assembled, translation by the ribosome would then move into the quaternary protein conformation to form megazord protein that's going to break down arsenic. But then you look at these other ones and it's like, oh, that's like 4 operons for this one, and then 2 different operons for this one. They kind of look like they would be on multiple different chromosomes or at different spots in the DNA. These are bacteria, so they don't have chromosomes technically. But okay, but there's multiple. So A looks pretty nice. E. coli only has 1 and multiple operons. I mean, I like A. If I'm going fast, that's a good answer. Both of these contain the arsenic gene that codes for a repressor. We don't really know anything about a repressor. It says repressor there. That's the vocab word I'm zooming in on. If we remember that from lac operon, trip operon. I'm like, I don't see literally anything about repressor in here. Nothing about repressor. I just can't guess that. There's no reason to guess that. And A is just a better answer. C says both of them codes for protein that helps remove arsenite for the cell. O, Triticus E. coli contain the ARSD gene. So I guess you could look and see the ARSD gene. So that's true. E. coli has ARSD as well as ARSD in E. coli, which codes for protein that helps remove arsenite from the cell. Well, they both have RSD, so they're trying to get you there, but does it say anything about removing toxin from the cell into arsenite and removing toxin from the cell? The problem with C is that we don't know if that's what RSD does. Nothing up here says anything about RSD, so that would just be speculation. C is just not the best answer. It doesn't, there's no proof of that. Okay, and then the fact that E. coli does it more efficiently doesn't mean that it doesn't do it. So it's saying here they have more arsenic resistance. Just because actually this is a more complicated way to get arsenic resistance. So it doesn't mean that they have more. We could double check all of that, but honestly, you just have to, maybe you could then narrow it down. B is obviously trash. C is like, meh. D is okay, but A is just better. So when we're going through that, we're really gonna have to jump to that. For here, they're just trying to test your knowledge that like, okay, it's a double helix, it's DNA. We have AUG, so we have some U's in there. So this is transcription by RNA polymerase, making that mRNA to DNA here. So the process here is going to be a nice one. Initiation of transcription, I like that. Translation, yep, C is out. Alternative splicing, that's not what we're seeing here. That's the step that comes right after this. So if I'm going fast, I'm saying B, and that's nice. A says DNA replication. Again, like my brain just kind of skipped over that because I'm like, I looked at this and I knew it was transcription. This is a case where the question was so short and then the diagram was also so short. I just kind of went diagram first and then down to the questions because they're just asking for a definition. This is a straight definition question. They just want to know if you can kind of visualize this and translate that into words. Oh, we know the answer is D from my mistake there. Okay, again, this one, there's not a whole lot going on. So I'm just like gonna briefly look at this. I am seeing something that looks like, okay, we're building DNA here, right? Maybe we're building RNA, whatever. We've just kind of got template, growing, great, okay? Question, model provides evidence for which explanation in 5 to 3? Oh, okay, right, all right. So now I'm looking at the 3, the 5, and then the anti-parallel nature of DNA, 5 to 3. So obviously we're going in the right direction here. We're moving over to synthesize that. So now we know we got an enzyme. It's probably DNA polymerase 3, and we're adding more nucleotides. So they're asking evidence. This is a provide evidence question, okay? So it's not asking for what's true. This is asking what does this model give us evidence for? And it says the word why. So like if you're underlining this thing, you're writing evidence why, okay? We know that it goes synthesized in the 5 to 3. You walk on 3 to 5. You build 5 to 3 because 5 to 3 works for me. If you're doing the work, you're building 5 to 3. You're walking 3 to 5. Okay. I like A. Seems really nice. Okay. If they're using that vocab word anti-parallel, pretty nice. Okay. But we got to double check. Thymine and adenine would not bond properly if the strand grew from 3 to 5. Maybe. I know that the other stuff wouldn't really work. Possibly that's the case, that like they have to line up like that. And if they're going like that, it's just not going to work, but your thumbs go together really nicely like that. Possibly that's good. We'll see if there's something better because A and B are okay. C, the translation of mRNA. Well, we're not talking about translation here. The growing DNA strand must also grow in the 5 to 3. I don't like that one as much because it's talking about the ribosome, and here that's not what we're having. So like maybe again, and then we just want to see if one pops up. Okay, so D, then I'm scanning to D. Phosphate group forms a covalent bond with the oxygen atom attached to the 3' carbon of the growing strand. Now, the phosphate group down here, 3 to 5, 3 to 5, we have a phosphate sticking out over here on the 5' end. We have the phosphate sticking out over here on the 5' end. Okay, so this phosphate sugar base kind of setup, phosphate, and there's a sugar that's not really pictured, and then the base that's sticking out over there. And it says the phosphate group is attached to the 5' carbon, and that forms a covalent bond with the oxygen atom of the 3'. This is going to be where the attachment is occurring. Now, I know the answer is D. We all saw it. Okay. Here's how we kind of describe this one. Okay. And again, this isn't always the case, but if you are rushing and you just have to make a guess, the longest answer here is the one that's correct. Just saying. Okay. A is just kind of too simplistic, I guess. Like I like A. I can understand why we would put that, but you know, the B, thymine and adenine, that's not really what's going on here. Those hydrogen bonds in the middle, those are just weak hydrogen bonds, that's just magnetism. Okay. So when we're talking about actually putting these things together, that covalent bond on the phosphate sugar backbone, that's what is necessary for that to occur. So that's why D is going to be our correct answer here. That is a toughie. Okay. Honestly, that's a toughie. But yeah, there's the right answer on that. But it is based on the DNA polymerase making the covalent bonds, and then those hydrogen bonds are just going to kind of like naturally occur between the A's, T's, C's, and G's. So 8, oh gosh, yep, we have no idea what that is. This is gel electrophoresis, which we have to discuss, but you know, there it is. Let's see if we can get a good answer. Which of the following statements best explains the pattern in the gel, regardless of size? So this is where we're not going to be able to cover everything, but maybe we can try and figure it out. Okay, we have wells, we have 1, 2, 3, A, B, C, D, E. We have the positive, we negative. Molecules A and B are positively charged. Molecule A is smaller than molecule B. Well, listen, we know opposites attract. Okay, so A is very close to the positive. So A and B, you know, wells, that's where these DNA pieces start. So I can just like kind of tell you that like that's gel electrophoresis. We put DNA in these wells. A well holds water. Okay. So it's like a little bucket, literally like a bucket in a gel. And then we electrify it. And the things that go towards the positive are going to be the negative ones. Okay. And the things that go towards the negative are going to be the positive ones because opposites attract. Okay. And DNA is charged. It's a polar molecule because almost everything is, except for fats, lipids, and yeah. So polar molecules charged, they're going to move. So molecule A and B are positively charged. No, they're moving towards the positive. So we know they're negative, and we know these guys are positive. So A is out. Molecules A and B are positively charged. Nope, B is out too. Okay, so C, again, this is one of those questions where you got to kind of sort through. Once you know they're negatively charged, you automatically know the answer is C or D. We just got to see if molecule A is smaller or larger than molecule B. Okay, well, which one moved farther? Molecule A moved farther. So if you are being dragged through a gel, which molecule is going to move farther? It's going to be the smaller one. Okay, the big ones, there's more drag, there's more resistance. Okay, like how long does it take a humpback whale to move versus, you know, like a little shrimp? Okay, shrimp goes fast. Okay, now yes, the humpback whale is bigger, so I don't know if that's the best example, but let's say if you're trying to drag a fish net through the ocean, what's going to move faster, small fish net or big fish net? Okay, small fish net. So A, the smallest, moves the farthest from the well. Okay, that's why molecule A is smaller, and C is going to be our correct answer here. Great. Gel electrophoresis. That's how we separate DNA of different sizes with electricity. Ooh, the enzyme lactase. That just like jumped out at me. I don't know why. There it is. And then we have a map of the Earth, and got to go to the question. Lactase persistence, mutations. All right, so we're talking some evolution here. We're talking about the lac operon and breaking down lactase, which is like, yeah, okay, great. Lactose intolerance, lactase persistence. And so let's look at the questions. Which of the following best explains the distribution of lactase persistence? Best explains. So I'm looking at this going like, distribution of lactase persistence. Well, where is it common? Is the black parts. Where is it not common? The white parts. So it's very common up here. You would just have to kind of look like, oh, this is Africa, this is Asia, India, and the Middle East, and then, you know, your basic geography over here. Guess you got to have taken AP World, kind of. And then over here in the Europe area, especially England, is where we have a lot of that lactase persistence. So the key thing I'm going to write down in my notes here is lactase persistence is not intolerance. Okay, so intolerance, no milk, and then persistence, milk. Okay, so A says lactase persistence developed because people were malnourished in Europe. Maybe, probably, I mean, B, lactase persistence alleles are present in all human populations and are expressed. Oh my God, come on. Okay. No, we see it right here. It's literally like it doesn't exist in the white areas and exists more in the dark areas. So this like B thing is just garbage. Okay. C, mutations conferring lactase persistence likely arose in different geographic areas. I don't know. You can kind of see that, but in general, it kind of looks like, okay, it originated up here in Europe and then it just kind of spread. And if we kind of know that Europeans colonized areas of Africa, we know that that's why it spread there. So it looks like it did originate in one place. So this arose independently means in multiple different places. I'm not loving that. D, mutations cause lactase persistence are detrimental to humans and will eventually disappear from the gene pool. Has this graph doesn't provide any evidence of that. It just has nothing to do with this graph. So like that's a distractor. I don't really like that. So what that's coming back to is my best answer now. As I'm scanning back, I'm like, C was total trash because it's I knocked that one out. B, and again, you know, you're writing this stuff, you're crossing these things out, so we know we don't waste our time going back like, which one did I eliminate? I eliminated C. Okay. B, lactase persistence. Yeah, we knew B was complete trash, and so it's between A and D. And you know, D is just not great, and A is certified okay. So again, they're asked, oh, and C is C wins. Nice. Okay. Arose independently in different geographic areas. Okay. So they are saying that it did originate here in this West African place. Okay. Where they must have relied on milk production. Great. Okay. Now remember, when we get one wrong, do you see how excited I am and how interested I am to learn? That is key. We don't have to get 100% on this AP test, right? And right now when we're practicing for it, every time you get one wrong, that's interesting. That's like, oh, cool. That's an opportunity to learn. That's how you have to feel. That has to be your automatic response every time you get something wrong. Okay. 12. All right, great. I'm seeing topoisomerase helicase. I know we're talking DNA replication. Hell yeah. Okay. Going down here and the answer's DNA replication. Great, great, great. So we have cannot occur, will be initiated immediately, site. Okay, so it looks like some sort of, this is a malfunction question, okay? I think, so it says, which of the following correctly explains where DNA replication will begin? Okay, we gotta take a walk from 3 to 5, which means that we're gonna build from 5 to 3. And on the top here, walking from 3 to 5, we're gonna build 5 to 3 there. And so we have a replication fork. It's not very open here. It looks like they should be separated a little more. We need some single strand binding proteins in there. But it's saying where DNA replication will begin on the strand oriented 5 to 3, reading from left to right. Lord, this is just a terrible freaking diagram. But I think that they are talking about 5 to 3 is this one. Your 5 to 3 ends up being this top one here, where the RNA primer is already present. And we can kind of see that it's going to go into the replication fork, because 3 to 5 means that this RNA is 5 to 3. 5 to 3 works for me. Take a walk from 3 to 5. So the top is going to be the leading strand, where we are replicating into the replication fork. Again, this requires a pretty strong knowledge of DNA replication, straight up. This is not one that you're just going to be able to guess or figure out. You got to know DNA replication. So let's say C looks really strong. DNA replication initially immediately to the left of the RNA, right? The primer is on, but then DNA polymerase is going to continue right there. Okay. Yeah, and I really like C. Honestly, it just jumps out and it's great. I don't like B, that why would it not occur anywhere? D, topoisomerase, we know that, no, we need a primer and it can't go through that spot. And then I'm just double checking on A, randomly initiated. That's just not how it works. You know, you got to start with a primer. As long as you know primer, then you're going to land on C. Nice work. Okay, then we're going to jump to 14. And we have, great, gametes, meiosis, cool. So non-disjunction, non-disjunction. My eyes just gravitate to like, okay, the beginning is part. So obviously we're talking about non-disjunction. Okay. That's a failure of chromosome separation in anaphase. Okay. So we got anaphase. Cool. 1 or 2. So now we need to decide whether it happens in 1 or 2. Well, this is the first separation right there. And then we have the second separation right there. So what I'm seeing is a problem right here. Okay, that must be in anaphase 1. Anaphase 2 happens over here. So this looks fine, but the problem was early on in anaphase 1 to go from metaphase 1 to anaphase 1. Then we're in metaphase 2, and then moving on to anaphase 2 and telophase. So we know we're talking about anaphase 1. All gametes will be normal. Hell no, that's got to be B. That's just easy to rip through. Okay, you got to be comfortable with those diagrams. Great. Then, ooh, a tough one. Let's go to the question. Oh my gosh, yes, please. Justify the claim that phytochromes regulate the transcription of genes leading to the production of cellular protein. So I'm slowing down on this one because we've got to know which ones to slow down on and spend some time on, okay? The transcription of genes leading to the production of certain cellular proteins. My gosh, they're going to use all these fancy things. We're just making a freaking protein. It ain't that complicated. Here's the nucleus. Okay, not so scary anymore. This happens, this happens, this happens. Okay, look, we got red light, and then we make a protein. It's not really that complicated here. So let's see what they're actually asking us to look for. When inactive, or this is a note-taking one, when inactive phytochrome PR is activated by red light, it's transported into the nucleus where it binds to the transcription factor that stimulates transcription, ultimately leading to protein production. Okay, so gosh, that seems like what's going on. We had red light, and now it's active right there. And then in the end, when it's active, do we see transcription or no transcription? I can't really tell. Maybe it's in the question here. It's definitely not. Oh, these are 2 different diagrams. It's not one giant cell. Here's one diagram, figure 1. Here's another diagram. Okay, all right. So this first one, exposure to red light, right there. Second one, exposure to far red light. And so A, which is the longest answer, notice the longest answer, okay? When inactive is activated by red light, okay, so let's say the red light one versus the far red light. Now I know I'm looking between those 2 different things. I'm circling this, far red versus red. A is red light, boom. Okay, see, just take your time and then you start to get oriented. Don't ever be scared of this stuff. Get interested, get excited. Red light means that transcription, game time, right there. Definitely looks like transcription's happening and we're making cell proteins with red light. So I really like A. We can keep looking at the other ones. Far red light activates, causing it to travel to the nucleus, or it binds to promoter, stimulates transcription. I know far red light, literally it says far red light, no transcription, no transcription. Like you just, that's a simple thing you're trying to look for, okay? Are we making a protein or not? In the first one, red light protein. So I like A, but again, C and D, they're shorter answers. And A just seems so clean. And B seems like the opposite of A. B is clearly wrong. I'm like an A. These PFR not activated by red light, causing it to bind a promoter. And why are we talking about MYB? Like what? That is nothing. MYB is at the very end here. That's not what's reacting to red light. They're just distracting you. And then PF3 binds the promoter only in the presence of red light and PFR. Okay, hold on. PF, it's there. No, PIF3 is bonded both times. Here it is. Here it is. Okay, so PIF3 is not any different between the 2. So A is really strong. Okay, cool. A toughie that we can work through. Now an easy. So I'm just going straight to the answers. Plasmids are circular. Plasmids are single-stranded. This is a memorization thing. Plasmids are circular. That's how bacteria keep their DNA, prokaryotes, circular. So I'm not even looking at C and D. double-stranded, single-stranded RNA molecules. Plasmid is double-stranded RNA, or sorry, double-stranded DNA. It is plasmids are DNA, period. So double-stranded RNA does exist. Single-stranded DNA does not. Single-stranded RNA obviously exists, but we know plasmids are circular. I'm not even going to read anymore. Maybe I'll get this wrong, but B, okay? If you have certain things memorized, that's why it's important to just know the basics of biology and to continue to study that. Okay, 20 here. Process represented figure 1. The modification of protein to produce, okay, all right. So we're making protein, like introns, exons, that is RNA splicing. Okay, so this one is based on your knowledge of RNA splicing. I don't talk about a lot. It's the step between transcription and translation when making a protein. You make your RNA, but then you got to like modify your RNA a little bit. You got to like cut it up and splice it a little bit. As you see right here, these exons are what is going to go be translated, whereas the introns are taken out. In, out, like in and out burger. Ins goes out, exons exist. Exons stay. You might think exons exit, just, you know, in and out, in and out. Okay. The exons here with the poly A tail and the 5' cap, bing, bang, boom. Which of the following best explains the process? If I'm looking, if literally anything says splicing, it doesn't say that. But we have pre-mRNA and mature mRNA. Well, I like D. Okay. You got to kind of, this is a straight up memorization thing. We got to know that this is just the step between transcription and translation. That's what it looks like. Okay. Taught it to you right there. So we can look at others, but D looked really clean to me. Another memorization problem that you just got to know your stuff. Okay. 22. This should look very familiar as protein synthesis. So which of the following best? Oh, I'm seeing insertion deletions, mutations. Great. So let's look up here, and they're even highlighting it for us. The DNA said CTC, and then the DNA in this one says CAC. We know it's a mutation. It says mutation. The first one is normal. The second one is sickle cell, and the change is right there in the DNA from thymine to adenine. That's what changes the DNA to the RNA, glutamine to valine. And what happened is we just had a simple substitution. We'll substitute there. Okay, that's straight up definition. You just got to be able to read that. Okay, so I'm feeling really strong about substitution. All right, then the last two. Great work, nice focus. Imagine you just got to do twice as, you know, we did the evens, right? So you got to do 4 times as much of this. So, you know, just think about the mental energy that it takes to do this. You kind of practice doing all 26 of these, sit down, see how much time it takes you, okay? And see and focus. Recognize your brain needs to do twice as much of that. With a good night's sleep and good food, you take care of your brain and you're able to do this, okay? Then you take a deep breath, maybe you stretch, relax as you're taking this. As long as you can get 26 problems done in under 40 minutes, you're looking pretty good here, okay? So, synthesis, right? We're talking transcription, gene expression. So this is our first step here. We open up the DNA, RNA polymerase, great. Which of the following best explains what strand X represents? Where's strand X? Okay, strand X is the DNA. Is that like the DNA? Yeah, strand X is DNA. It ain't no RNA, boom. The D is wrong too. B, the coding strand, because it's read the antisense strands because it's serving as a template. Whatever this antisense things means, I'm telling you that bottom strand X of DNA, that is a template. So I like that. B says the coding strand. I wouldn't really call it the coding strand because it's being read 3 to 5. Coding strand would be more of a reference to mRNA. I like that more with ribosome. So again, they're throwing vocab at me that literally I haven't even heard this vocab. Okay, I'm gonna be honest. I'm not perfect. I don't know everything there is to know about AP Bio, but I do know how to take tests and do your very best. Okay, nobody's perfect. No one taking this test needs to get 100%. Neither do I, but I do know what a template is. That's how I can still get the right answer on C. And then we get to our last one. Wow, why is this font so giant? Yeah, okay. So we're looking at inhibitors and prokaryotic eukaryotic cells. So some cells are going to length of polypeptide chain. Oh, so making proteins here. So, gosh, control, experimental. All right, so all of these have the control on the left, and then they have the experimental on the right. Again, what I'm trying to do is find what the heck is the actual difference in these questions. All of them, the left one is exactly the same. Don't focus on it. On the right, we have, all right, so here, the prokaryotic goes up, eukaryotic stays low. Up, low, it's the opposite, and then C is the opposite of D, right? So A is the opposite of B, C is the opposite of D. We just got to figure out which one goes up, which one goes down. Is it the eukaryotic or the prokaryotic? Okay. Our x-axis is almost always time. That's what we're seeing here over 5 hours. And the y-axis is the length of the polypeptide chain. So I'm writing it right there. I'm writing protein length and then time. Okay. So all I got to do when I'm going up here, I'm looking, listen, is it eukaryotic or prokaryotic? We got bacteria making long proteins, or do we got eukaryotes making long proteins? That's all I'm looking for. Passage, antibodies. You got to know antibodies are proteins. In separate dishes, radioactive, specific, synthesize, identical. Which of the following sets of graphs best summary? Okay, the procedure was repeated, added each, and the translation was monitored again. The specific gene was engineered to synthesize identical polypeptide chains, and the translation of the gene was measured. Okay, so gosh, this is so interesting. Inhibit the movement of substances through the nuclear pores of rat liver cells. And that first vocab word, nuclear. So the one thing that they are testing in this thing is, do you know, prokaryotes don't have nuclei, period. Okay, so this has everything to do with whether you have a nucleus or not. Okay, regulate the passage of substances. Antibodies inhibit the movement of substances through the nuclear pores of rat liver cells. Okay, so if you have the procedure is repeated with the inhibitor. Okay, so what I'm writing down here is inhibitor, the MAB inhibits. I'm going to draw an arrow right there because what that means is it said no substances into the nothing in nucleus. Okay, this is the last one, but I'm not rushing. Okay, nothing in nucleus with the inhibitor. Nothing in nucleus means no protein. Okay, no polypeptide, no protein. So to me, I'm thinking like that's going to block out my eukaryotes, but my prokaryotes should be totally fine. Okay. So in the control, there's no inhibitor. Everybody makes proteins. Bing, bang, boom. Make your protein, make your protein, make your protein. Over here, it's kind of like the prokaryotes should have no problem. The eukaryotes, if you're blocking something from getting into the nucleus, you can officially block those eukaryotes. So it's definitely A or C. I don't have any reason to believe that they're going to make some and then like slow down. I don't think your eukaryotes are going to be able to make any. Okay. I just have to make that solid guess. And there it is. Okay. A couple of different steps on that one, being able to read the graph and see what the X and Y axis are, but then also just knowing that nuclei are in eukaryotes. And then I had to go over and write down and make sure I didn't get confused about the inhibitor. But this one did require me to go back to the actual question and really look at what are they saying and draw a little diagram. Okay. Inhibitor means nothing in the nucleus, which means no protein. Okay. And that I can put in parentheses is only for the eukaryotes. And I could draw a really strong line because it tells me in my key here that the eukaryote is a strong line. All right. Well, thanks for freaking showing up and working on this stuff. That's a good one.